realize that many of you haven't taken the time yet to learn all the words that we talked about yesterday, because you were probably studying for the quiz that is due tonight by midnight. So I'll try to remember to repeat the definitions of the words as we go along. So let's start with this. Okay, so Mendel did his experiments, and remember we said like 1857. And again, it's not that I am interested in you absolutely memorizing that year. What I'm interested in is that you remember the time and sort of what else is going on in science at that time, right? Especially biology. And Mendel bred hundreds and did experiments on hundreds of peas. And we already went through right? He took true breeding parents, so those would be homozygous, a pea generation, and he took purple, and it's unfortunate that P is for purple, and we put a little X there for he crossed those with white flowers, right? And we said yesterday that the first generation, all the offspring were purple, right? These are true breeders, these are true breeders, these are not. These are going to be hybrids. And then when he let the F1 generation self-fertilize, which we usually express like this, an F1 crossed back on itself, an F1. If we were talking about an animal, like a fruit fly, for example, it would be a sibling cross, right? Which we obviously can't do in humans or larger animals. But in plants, because they have both angiosperms, in particular, flowering plants, they have both male and female gametes inside a single flower. You just have to let the flowers self-fertilize. In the F2 generation, remember he saw the dominant allele again, purple, and then the white reappeared. And remember, I think I told you yesterday that it was the numbers of purple to white that was what was important and helped him figure out his law of segregation, his first law. There were 3/4 the amount of purple to 1/4 the amount of white flowered plants. Okay, so now, not until later, early 1900s, and I didn't tell you this yesterday, but sadly for Mendel, he published 2 or 3 papers with his 2 principles, segregation and independent assortment, and then another follow-up paper, I think. And then, unfortunately, because he was using math, and so he was talking about ratios and percentages, and at this time, biology was still very much an observational science, and not much data, specific data, numerical data attached to it. Oh, who was that just entered? That, and because remember he was in Austria, or what was then Austria-Hungary, he was writing in German. And quite honestly, the language of science at this time was French, predominantly, and then of course, like naming things, of course, would have been in Latin. So French or English and Latin, but not German. So Mendel's work did not get a lot of buzz, right? Especially because we didn't have as many forms of communication as we do today. So it wasn't until the early 1900s that we say some people rediscovered Mendel's work. And 2 important people to do that was one guy named Punnett and the other named Bateson. And they were interested in if this extended to other flowers, other plants. They were just like, wow, this is really cool. And Punnett in particular was like, I wonder if there's a way we can mathematically predict what the offspring are going to look like based on sort of Mendel's principles. And so he is the originator of the Punnett Square, which is basically It's almost like a probability chart or a prediction of what possible alleles or what possible alleles could be in a gamete, and then what are all the possible gametes that could join together to form new zygotes of the next generation. And so we call these Punnett squares. And when we have one characteristic, one character or trait, like we do in this example that we're doing, when we have flower color, that's the only thing we're looking at, we refer to this Punnett square as one for a monohybrid cross, because it's just one thing. And in our case, like I said, it's flower color. And we already know that the two alleles are purple and white. Okay. Okay, I'm going to put up a new board to continue here. And so in a monohybrid cross, we would make a Punnett square that has 4 subsquares like this. Okay. So it's 4 boxes. And then many times you'll see a diagonal line there and a demarcation up here at the top. This is for which parent is the male parent and which parent is the female parent, because sometimes that matters. And then these lines often are extended like that, because what we are going to place across the top and down the side in these extensions, what we're going to put there are possible alleles in the gamete. So we usually just shorten that to say the gametes. Now again, I'm going to try to go really slowly so that you guys can follow along. So I'm going to change my pen color a little bit, and I'm going to go back to Mendel's experiment when he had his P generation of purple flowers, purple flowered peas crossed with white ones. Okay. And so Punnett set out to say, okay, there has to be a way that we can designate purple very quickly and white very quickly. And how we do that is we choose, in plants in particular, we choose a letter of the alphabet, and we choose, you'll hear me sometimes call this the symbol for the character, or for, I should say for the character or for the allele. We choose usually the first letter of the word from the dominant allele. And this usually works unless our 2 alleles both begin with the same letter. And then a lot of times people will use the first 2 letters. Okay, but in our case here, in our example, so we already know what is dominant between purple and white. How do we know that? Because when purple gets crossed with white, remember the F1 generation, they are all purple. And we said specifically when we talked yesterday about the Mendel's first law, the law of segregation, that the dominant allele is the allele that masks or hides the recessive allele in the F1 generation. So that's key. So because they are all purple, that makes purple the dominant allele. So purple is the dominant allele. In this case, it's not like in peppers that we eat, but it is in flower color in peas. Okay, so they're all purple. So purple's the dominant allele. So we're going to choose to represent purple then up here. We are going to choose the capital letter P. Oh, I should probably make that a different color. We're going to choose capital P to designate purple. And thus, we are going to choose the same letter, only the lowercase of that letter for the recessive allele. And this is where people usually run into trouble because they want to make it a small w. They want to make it a lowercase w or a little w, as we would say. But no, you have to keep it the same letter. When the letters look the same when they're printed, I usually try to make the recessive look cursive so that you can tell the difference clearly between the letters. Okay, so questions so far, because this is key. If you don't have this, then we're in trouble. So I'm gonna stop share and ask. Anybody have, how do I see you again? Gallery view, there you are. Anybody have any questions about that before I go on? So for the dominant allele, we use the capital letter. It's usually the first letter of the word for the dominant trait. In this case, it's P for purple. And for the recessive allele, we're going to use the lowercase of the same letter. So that's going to be a lowercase p. My little table here. Okay, so if you guys don't have any questions about that, I'm just going to go on. Oh, yikes. My whiteboard disappeared. I forgot that happens. Okay. Okay, so let's go back. So we have our P generation, and we have, so that's true breeders, purple crossed with white, flowered pea plants, right? So the characteristic is flower color. Alleles are purple or white. No, and we said for purple, we're going to designate that as a capital P, and for white, we're going to designate it as a lowercase p. Now, these are parents. This is P generation. So we need to account for both of the alleles because remember, pea plants are diploid. They are 2N. So we are going to put a second capital P here and a second lowercase p here. These are homozygous because they are the same allele, right? It's kind of lame there. Let's look a little better. Lowercase p, like that, two little p's. They are homozygous because they're the P generation. They are true breeders. So now we can translate this to our friend, the Punnett Square. Put a Punnett Square over here. I'm going to draw it again with 4 boxes, put the diagonal. In this case, it doesn't matter which parent represents the male and which represents the female, the stamens versus the ovules. I'm going to put this parent, and I'm going to call it just the circle parent. I'm going to put that across the top. And so what I do is I take a anther, in this case, or a pollen grain, in this case, because it's a plant, could have either this capital P in it or that capital P in it. That's all that this parent can possibly give. This parent, the other parent, the white parent, put that one in a triangle, right? The alleles that they have to give clearly are only lowercase p or lowercase p. Try to remember, these are gametes. These are possibilities. This is like doing some probability, right? And in this case, they are the same, right? But if this is one capital P on chromosome 1, and this is the other capital P on chromosome 1, and I'm going to refer back to our drawings from yesterday. Remember when I had those chromosome 1s, and I had one that was plain and one that was stripy? And I know they were green. They're just black today because that's what I have in my pen, right? And I did this, right? So this is chromosome 1, and this is chromosome 1. This is what I'm depicting here. In this parent, which is 2N, in its gametes, it can put either this capital P, they happen to be the same thing, or this capital P from this chromosome. The other parent, same, this lowercase p or that one. Then to determine what the zygotes will look like, what the offspring will look like, that is when we fill in the boxes. Okay, now there's some rules about filling in the boxes. When you go to fill in the boxes, you always put the capital letter, if one is present, you always put that first, and then you put the lowercase letter. So I'm basically, what I'm drawing here is I'm saying, what happens if this pollen grain meets up with this egg? and becomes this flower in the next generation, this seed. And then I'm saying, okay, what happens if this pollen grain meets up with this egg? Okay, Aloma, I'll keep an eye out for you and then let you back in. That's fine. I'll see it when you rejoin. Okay, and then what happens if this allele meets up with that one, and so on. Right. So now I have all the boxes filled, and now I look at my symbols and I try to determine what that's going to tell me about the phenotype. So remember, when I have 2 capital P's, that's definitely purple. And remember, my F1 generation was also purple, and that's what I'm depicting here in these boxes. A capital P with a lowercase p, those also look purple. They only show the dominant trait. Yay. So next to the box, you would write for phenotype, all purple. And if I asked you the genotype, or what alleles do they have, you would write capital P, lowercase p. That's the genotype. Like that. Okay. When I ask you to solve a genetics problem, what I'm going to be looking for is they're usually worth 5 points, and each part of what I've drawn out here is going to be worth a point. Okay, not each part, not the chromosomes. But what I would expect you to be able to do is identify the parents and show me the cross that you're setting up. We call this the cross, right, that you're setting up, the fertilization that you're hoping will happen. Then that's worth 1 point. Assigning the correct alleles, letters to the alleles, sometimes is also worth a point. Setting up the Punnett Square is worth at least 1 point, 1.5 points, sometimes 2 if it's more complicated. And then this last part is the part that students mostly forget. This part is important because this part is the actual answer to the question, right? So if the question was, what will the F1 generation look like? Or what is the appearance of the F1 generation? The answer is all purple, but you have to show me your work in order to get all the points. Okay, so let's continue. Right, so we're still translating the purple and white flowers that Mendel talked about with his peas into, right, into first the F1 generation and now the F2 generation. Okay, so these are the F1, these are the F1s. They are offspring of 2 true breeders that have been crossed. So the F1 generation, that first filial generation that is all purple, they are also all big P, little p. So when we let them self-fertilize, that makes it looks like this. Yeah, so this is one F1 crossed with another F1. And that is how we produce the F2 generation, or the F2 offspring, by crossing an F1 with an F1. I should have written that here. Let me erase this part and just put above this maybe. How about that? Let's put above this F1 crossed with F1.
I have a question.
Yeah.
The F1 generation, is it just the top 2 or all 4 of the squares?
Oh, sorry. It would be all 4 of the squares, Kira. That's
okay.
a good question. Yes, all of them are F1. So it doesn't matter. I could cross this one with that one. This one, I could do any of the combinations because they're all the same, right? And in the F1 from 2 true breeders, you're always going to get all heterozygotes. So now I would draw a second Punnett square to show how I would get the offspring of an F1 crossed with an F1, which are going to be the F2 generation. And there, now we're going to see some variation among the offspring. So I'm going to move my box over a little bit to the left here. And I'm going to make another 4 square like that, because I remember we call this a monohybrid cross, right? One trait, we're just crossing flower color here. And now I would put one of the parents across the top, same as before. But now I'm going to use a capital P and a lowercase p, because this gamete from the blue parent could get either this chromosome or that chromosome. And now one of these is a capital P in the red area and one of them is lowercase p in that red area. Same for the other parent, right? We could get a capital P or a lowercase p. Now we're going to get different results when these form zygotes, which is what we're forming in the boxes, in the inside boxes, right? On the outside, remember, these are gametes. These are the results of meiosis. These are what is possible. And then now we're asking, okay, if this pollen grain meets up with this ovule, I would get this zygote. That would be its genotype, 2 capital P's. But if this pollen grain meets up with this ovule, I'm going to get that. Notice I put the capital letter first. That's so we can easily see the color. We can easily see if there's one capital P, we're like, oh, that means purple. What about if this pollen grain finds this ovule? Now, even though the lowercase p is on the top, we don't put it first in the box of the zygotes. We always put the capital letter first. So I'm going to put first the capital P and then the lowercase p. And then what if this egg meets up with, or this sperm meets up with this egg? I would have 2 lowercase p's. Now, capital P genotype gives me purple. So I would write over here purple, and we start to make tally marks. That one's purple, and then we cross it out. This one's purple, heterozygous, but it still has the dominant allele, so it looks purple. It's hiding the white allele, but it looks purple. Same for this guy. Looks purple, hiding the white allele. Oh, this is different. 2 little p's. The return of the white flower from the original parents. This is where we get that ratio of 3/4 to 1/4, purple to white. This is the same, guys, every single time. This is the same. If I start with true breeding parents and it produces an F1 and the F1 self-fertilize, the F2 will always be 3/4 to 1/4. I can only see white in the phenotype when I have 2 little p's. All right, questions, comments, worries, concerns about that?
I have one. Which were heterozygous again? The F2 generation?
So anytime when they have 2 different alleles, they're heterozygous. So the parent generation, the true breeder parent generation is always homozygous. Starting with the F1, the F1 are all heterozygous over here, right? Now, in my F2 generation, we would designate this first one that's capital P, we would call that homozygous dominant, because I have 2 capital P's. These 2 are heterozygous because they have 1 capital, 1 lowercase, and then this one we would call homozygous recessive. Homo for the same, homozygous, but it's showing the recessive phenotype. Better or worse, Kira?
Better, thank you.
Better. I know it's a lot of words, it's a lot of vocabulary, and I did not print the problems that I assigned that are at the end of this. Wait a minute. Oh, there. Okay, so let's try another one, and I'll go through it with you again kind of slowly, and you guys can ask questions just like before, just interrupt me. Let's try a different trait. Let's see, I'll pick on somebody from the audience. Let's see. All right, Jessica, you pick a trait from the list of Mendel's peas. We've got flower color, seed shape, pod color, pod shape, flower position, plant height. What's your choice?
Let's do pod shape.
Pod shape, she says. Okay, so I'm gonna give myself another whiteboard here and I'll go back to black. So she says, okay, let's look at pod shape. So according to Mendel, pod shape, we have two shapes. One is inflated and one is constricted. Inflated, so these would be the alleles, or constricted. Right, so the pod shape, that's the character, the trait, the gene that we're looking at. And then these are the choices. These are the alleles, the versions, varieties that are available. Okay, now we'll pretend we're like Mendel. We don't know which one's dominant and which one's recessive yet. So for 7 generations, we're gonna take pea plants that have inflated pods and breed them with themselves so that we continually get all inflated pods and we make true breeding parents. So our pea generation would be inflated, true breeders, homozygous, crossed with constricted. And I haven't given them symbols yet because I don't know what to pick until I know who's dominant and who's recessive, right? So I would cross those. I would look at the offspring of my F1 generation. And if I told you all are inflated, which I think is correct, give me a second here. Yeah, all are inflated. What would you tell me about what letters I would choose and how I would now go back and assign alleles for the parents, and then how I would make my Punnett square? Tell you what, I'm going to give you guys like 30 seconds, a minute to think about that. I even put you on a timer so that I don't start talking before I said I would. And then I'm just going to start filling them in, and you can tell me, you can think about if that's what you picked or not. So first thing we have to do is assign symbols or letters. And you want to start with assigning letters for the P generation. And then if you can, try to make your first Punnett square. Okay, so did you pick, they're all inflated. So that means inflated is the dominant allele. So I would pick capital I for inflated. And in the beginning, when I'm first doing a Punnett square with new characteristics that I don't know very well, I do this. I write capital I blank is inflated, and then 2 lowercase i's is going to equal constricted. And that helps me remember what my P generation is. So now my P generation, so they would be true breeders. They would have 2 copies, inflated, crossed with, constricted, like that. They're supposed to be eyes. And let's make our Punnett square over here then. So it's monohybrid cross, so 4 boxes, and I would have this parent across the top, this parent across the side, like that. And then if this pollen grain meets up with that ovule, I would get that. And probably by now you guys can see they'll all be the same, right? So then I would write next to this all inflated as the genotype, right? And you could put 4/4 if you wanted, if that made you feel better to use the fractions. 4/4 inflated, and their genotype, they are all heterozygous. Right? Yes, no, maybe so. hopefully. Okay, now if I continued and said, what would my F2 generation look like and what would their genotypes be? Right, to get to the F2, remember we would let the F1 self-fertilize. So I would do this, F1 crossed with F1. And then usually right underneath it, I write the genotypes for the cross, right? So I would put heterozygous like that. And then I can make my Punnett square for my F2 generation. Right? So I could do this. And again, I would put capital I, lowercase I, capital I, lowercase I, because I'm taking, if this is, we'll make this circle parent, right? And I'm putting that across the top, one allele in each box. Okay, now, capital I, lowercase, capital I, lowercase, and then two little i's. All right, so I'm going to open the participants and then I'm going to ask you guys to tell me. I'll just start at the top there. Andrew, what does this one look like? Let's see if I can unmute you. No, I get it. A lot of you just turn this on and then, oh crap. I think my content went away now. Ugh, yikes. All right, I'll just redraw that box real fast for the F2. All right, I have this. All right, so this parent, I'll take volunteers. Anyone want to tell me what that one looks like?
Inflated.
That one's inflated, correct. So I would write, because this is where I make my list of phenotypes over here, right? So I'm going to put inflated and a mark for that one. All right, what's this next one going to look like?
Inflated.
That one's inflated, yeah. And what about this guy?
Inflated.
Inflated. And what about this last one?
Constricted.
That one's constricted, right? Yay. So we've got 3/4 inflated and 1/4 constricted for our F2 offspring. Okay, this is not so bad, right? With me going through it with you and reminding you how to set it up. And then now you're going to have to do it on your own. And so if you look in the module, there is a handout titled How to Translate Genetic Word Problems into Punnett Squares. And at the end of that handout, I think, is the first set of practice problems. They are all monohybrid crosses. And I tried to be very specific with what you need to include in your answer for each of them. And I would really like you guys to give those a try before tomorrow, because tomorrow I'm going to add on some new exceptions to Mendel's dominant and recessive. And if you've practiced these, that would be great. If you haven't, I might put a few of them up and ask people to solve them with me like we did today, so that we can kind of see how you're doing with the genetics problems. You cannot wait to do all these at the end. I'm going to make them all due, like I said, on probably Monday before the quiz. Try to make you get them done before the quiz. And then, actually, I'll probably make them do Sunday night. And then Monday, if you still have questions, we can go over them so you'll be ready, better prepared for the quiz on Tuesday. So does anybody have any questions right now that you can think of? no, yes, no. All right then. Tomorrow we will, I'll take questions. If you have questions about the genetics problems, I'll be able to answer them. And like I said, I'll probably go through one more of them with you because I would imagine we'd have more people show up tomorrow since we don't have a quiz tomorrow. And then I will press on with lecture 3 or part 3 of this book. Oh, no, actually, I guess it's lecture 3, which is the dihybrid cross. So it's similar, but we're going to add more boxes. And then Thursday, we'll do the extensions. And like I said, then Friday, I'll host a Zoom meeting for anybody who has questions about the problems, and we can go over them together, the ones you're having trouble with. And then that'll be it. There'll be no mastering homework from this chapter. You can certainly go to sure, Joe, I might make that just do Monday for you because like I said, I really want people to get it done sooner rather than later so that they have the practice in before the actual quiz and before they have work to turn in. Okay, so I'm probably going to make it do Sunday, but just remind me, send me an email after I put the dates on the homework, on the problems, the homework. There'll be genetics problems. Just send me a private email and I'll move yours to Monday. That's fine. Oh, I want to stop recording. Okay.